All right. So in your differential expression analysis, basically, after you get the RNA-seq data, you will have a FASTQ file. When you run Salmon or star-seq followed by RSAM, you will get an expression index value. And that's estimated sample by sample. Right? It's normalized together, but every sample will have a file and you put them in a directory. And then when you run DE-seq, you specify these samples are my treatment and these samples are my control. And then it will give you the level of differential expression. At the end, you also want to kind of show people the level of differential expression in a more visual way. And one of the really nice things that computational biologists did for biologists is this heat map idea, which is really very intuitive. So supposedly you have two conditions. This is only two samples, and you can have a legend. So each row here is one gene. Each column here is one sample, and the color in this case indicates the level of gene expression in that sample. This is supposed to be much, much better than looking at a big table of numbers. So most of the time we use TPM, or you can also use log TPM to present the level of expression of some genes across many samples, right? But you can imagine if you have a big matrix of numbers, it's very difficult to make sense out of the data. So people, computational biologists, this is Mike Eisen, back then at Stanford and now at Berkeley, he developed this heat map approach where you can visualize the level of gene expression. Of course, in the left figure, the genes are not ranked in any particular order, which is really hard to see. But if you look at the right side, there are many samples and also there are kind of different rows. So the rows are the genes and columns are the samples. You might notice that in this group of samples have, by the way, maybe red is upregulated and green is downregulated. We need to provide a legend so people understand, you know, what's the level of up versus down. But you can imagine this would mean that a group of these samples are really high on these genes and these genes, but lower on other genes. And then the remaining sample are much, much higher in these genes, but lower in the other group of genes. And so that's really helpful for people to understand the data much better than a big table of numbers, right? Imagine doing this for hundreds of samples over thousands of genes. It's very difficult to see, but once you draw a heat map, you can actually understand the data better. So most of the heat map convention, people use columns to indicate sample and the rows to indicate gene. And I hope you can see from comparing the left figure and the right figure, the order of the rows are very important. You know, this right figure, if I don't order it, or if I order it by alphabetical order, it will be a big mess of black things. You will not be able to see much. But now that we have ordered them so that the genes that have kind of similar expression patterns are put together, you see them much better. And so how do you order them? That's related to clustering. So, so this is, say, the expression data, right? Each row is a gene, each column is a sample, and we can cluster the samples in both column or rows. If we cluster the samples by genes, we are essentially asking, do we have genes that have similar expression across many, many samples or conditions, right? Then they should be put in adjacent rows together. We can also cluster by samples, which is clustering by the columns, which we are asking whether some samples look more similar to each other across all of these genes compared to other samples, right? So both give you some meaningful result. And so the clustering method that we're going to show you, you can either cluster by genes or you can cluster by the samples. And so basically you're clustering the vector, whether it's a column vector or the row vector, okay? So in order to do clustering, we need to, the reason we want to do clustering is because we want to visualize the data. For example, differential expression. We want to get a general idea of the characteristic of the data, and make a generalization about the gene behavior. And so for example, you might see some situations where you treat the sample with a drug and you followed over time, maybe 3 hours, 12 hours, and 72 hours or something. And you look at, if you look at every gene separately, it's kind of very messy, but once you group the samples, or cluster the genes, you might be able to say that the way the cell responds is this group of genes initially go up a lot and then gradually go down. But then there's another group of genes initially don't change much, but gradually go up. So you can describe the whole group together, right? So this is an example. You can describe the group behavior. And so this red and pink and blue genes, they have very similar behavior, so you can describe them together. We can also use this approach to classify samples. We'll have a lecture directly about the sample classification, but you can imagine if you can cluster the samples, you might find out, okay, there's some, disease versus normal, and you profile many patients, you can see, oh, all the normal normal samples are clustered together and all the disease samples are clustered together. That's also very useful to learn. In order to decide whether our cluster is done well, we first have to decide how to calculate the distance between samples. Very often, you can calculate, supposedly we have, let's look at this situation again. Supposedly we have a RNA-seq experiment. Each row is a gene, each column is a sample. Very often people use the log TPM value in here to represent. Remember, TPM is what you use for your gene expression, but very often people use the log TPM because there are some genes that are expressed much, much higher. So you kind of want to adjust the noise a little bit. And so you can calculate the log TPM on every gene, and then you can then compare the two samples to see whether they look similar or not. In order to estimate that, you can use, for example, Euclidean distance, right? Which is, for every gene, you have one number here, another number there, and you calculate their distance. And then you look at the next gene, you look at their distance, and so on. You can look at their Euclidean distance, but sometimes knowing that the expression level for TPM, sometimes they are not exactly comparable. For example, if gene A has a TPM value of 5.8, gene B has a TPM value of 5.5, we don't necessarily know that 5.8 is better than 5.5 because it depends on the GC content, the other gene lengths. We think we try to normalize. If you have a TPM value of 10 versus 5 on two genes, clearly they are different. But if it's like 5.7 on one gene and 5.8 on another gene, you sometimes are not so sure. So very often we want to learn about the general behavior of the gene rather than the absolute expression level. And so people also use the correlation. So this is a Spearman correlation or a Pearson correlation. And so you can correlate the gene expression of some genes across all the samples together. And a correlation is usually giving a range between -1 and +1. And you can use it to look at their distance by doing 1- correlation. Basically, it means that if these two genes are completely correlated, their correlation is 1, their distance is 0, meaning that these two genes are extremely similar to each other. Whereas if these two genes are completely opposite, their distance is 2, which means they're very, very different. Does that make sense? Some people also use an absolute value of correlation, so the 1- absolute correlation, which means sometimes even if two genes are completely opposite, maybe they are also related. And you can also use that as well. And in order to decide whether you are doing a good job in terms of clustering the samples well, the goal is we want to maximize the between cluster distance. You want to, different clusters to be as different as possible, but within cluster, the samples to be, or genes to be as similar to each other as possible. Okay. Another way to evaluate the clustering quality is to look at cluster stability. You want to ask, for example, see whether the clustering give you the same result if you mask out some data. For example, if you take out one sample or a few genes or maybe a few samples, the remaining data still cluster together roughly the same, that means your clustering is pretty stable. Sometimes people also try to introduce a little bit noise in the data. So for some random genes on random samples, you add a little bit noise to this. And if the sample cluster or the gene cluster still maintain the same, that's also pretty good. And the third is if you change the clustering algorithm parameters a little bit, if the final clustering results are also pretty stable, that will give you an idea that this is a pretty good clustering result. Okay, and also in order to reduce noise, a lot of people, they might first select a subset of genes for clustering. For example, instead of using all the genes, like 20,000 genes in the study, you might want to at least use some genes that are more or less differentially expressed within all the samples, at least having variabilities. If this gene is the same across all of your samples, putting them in your clustering is not going to help very much. Okay, so that's the idea. You can also select genes on certain pathways. So in general, if you select good, differential, for example, the most variable few thousand genes, they will probably give you a more stable clustering result. So the very early days when people were doing clustering is using hierarchical clustering. And what I'm showing here is we're trying to cluster the rows. We're trying to figure out which genes are similar to each other. And each dot here indicates one experimental condition. Maybe you treat the cell with some drug and you observed gene expression over a time point. And you want to ask what group of genes have similar behavior. You can do hierarchical clustering also on the sample level, but it will be very, very similar. Then you would just look at the sample across all the genes. So let's look at how we calculate the hierarchical clustering by clustering on the genes. The first thing we do is we have to calculate the pairwise distance of every gene with another gene. So with gene 1, from the 5 numbers, we can calculate their distance. For example, in this case, based on the correlation of their log TPM value between gene 1 and gene 2, gene 1, gene 3, gene 1, gene 4, and just every pairwise. Okay, so that's n times n minus 1 divided by 2 comparisons, or the pairwise comparisons for the distance. And so for hierarchical clustering, what you do is you first cluster the samples that have the smallest distance, which means that the closest two together. And then after that, you try to look, you calculate this cluster, this will become a node, and compare this node's distance with every other node in the remaining cluster, or the remaining data. And so basically you use this cluster to calculate its distance with gene 3, the cluster distance with gene 4, cluster distance with gene 5, gene 6, gene 7, gene 8, and so on. And after you have calculated all the new pairwise distance, you again pick the smallest distance again to merge. And so in this case, in addition to 1 and 2, we decided 3 and 8 are also quite similar. You can see it's like this. And then this will form a new node, and you will calculate the new node distance to all the other existing genes or nodes again, and then again pick the smallest pair, the pair with the closest distance again to merge. And so in this case, you pick 7 that merge with the initial group, and then that form a new node. And every time you have a node, you calculate its distance to all the other genes or nodes. And then you just do this repeatedly until you finish merging everything. Yes, question?
Once you merge the cluster, you try to calculate the distance with that cluster of other groups. Which point in that cluster do you use?
That's a very good question. How do you calculate the distance between one node to another node, right? And this is called linkage. Is how do you calculate the distance from the newly formed node to all the other nodes? And there are a number of different ways to do this. One is called the single linkage. So supposedly you already have one node which has 4 genes and another node that have 4 genes, because it's in a node you can see here. This node supposedly have 3 genes and this node has 5 genes. So the way to do single linkage is you calculate all the pairwise, so the distance from a dark blue to the distance of a light blue, and you pick the closest. The complete linkage is you calculate all the blue, the dark blue to the light blue, and you take the maximum. And there are also implementations called the average linkage, which is you calculate all the pairwise and you average them. Okay, so these are different linkages. That is one important parameter you can specify using hierarchical clustering. And so the default hierarchical clustering in the current R function is using this complete linkage. Okay, so initially, when you have just 2 genes merging, it's very easy. You just do the 2. And then you try to calculate a node of 2 genes with another one. That's still pretty easy. But later on, you will have bigger and bigger cluster. Then it's by looking at, you know, all the genes in one cluster with all the genes in the other cluster and calculate their distance. And you either pick the minimum, the maximum, or the average. And that's different linkage. Yes, question.
Is it always pairwise, or can you have it be more than pairwise? Because I see in the bottom the dark blue, pink, and black clusters. It looks like there's 3 of them coming together, as opposed to just like two of the clusters coming together.
Oh, you always try to merge 2 at a time, and you always pick the 2. The reason that sometimes, so by the way, if you do this hierarchical clustering, the lengths on here indicate how different the things are. And sometimes you might see the edges all sticking together. That just means the next cluster to this one is quite similar to the existing cluster. Okay. And so, yeah, so you can specify. So one way to decide whether your cluster is stable is to try different linkage, you know, single and complete and average, and see whether you roughly get the same result. And if it is, it's a pretty stable cluster. Yeah, so there's a brain teaser. If we have n data points, in this case, n number of genes, how many internal nodes are there in the hierarchical cluster? If we have n genes in the genome and you try to cluster your hierarchical cluster, how many nodes are there in your hierarchical clustering? Total, yeah.
In case n-1 steps.
Yes, exactly. That's the right solution, because every time you merge 2 to form 1 node, next time you merge 2 to form 1 node. And so if there are n number of genes, you have to merge n-1 times until the end, okay? Another question, how many possible ways can you draw a hierarchical clustering if you have a number of genes? So let's see this case. We are trying to draw, so 1, 2, 3, 4, 5, 6 is 6 different genes. And initially, if you merge 1 and 2 gene into one cluster, you can choose to draw the 1 and 2 this way or this way. That's 2-way. And then say next time you merge gene 3 and 4, you can draw it this way or that way. And another one, you merge 5 and 6, you can draw it this way and that way. And then next time when you have this, you can move them this way or that way, right? So the total possibility is 2 to the n-n power is like crazy number of ways you can draw this hierarchical clustering tree. So a pretty good hierarchical clustering algorithm will try to do a reasonably good job so that 2 and 3, if it's placed like this, will look more similar to the 1 and 4. so that roughly similar things are still close to each other. But that's kind of the specific implementation details. Okay. So roughly you can do this, and in fact, very often you can see when people have a big matrix of gene expression, they might choose to do hierarchical clustering on both the genes and the samples. So you might see this dendrogram, this treegram, on both the sample size, sample level and the gene level, and it's all pretty good. All right, that's the very simple way of clustering samples or genes. Questions for hierarchical clustering?
How do you decide the final number of the cluster?
You don't. Hierarchical clustering is just from 2 genes at a time, 3, 4, you know, whatever, until everything clustered into one. You don't decide a cutoff, right? And hopefully the things that are close to each other will be more similar to each other, right? But that just gives you a rough idea.